Acetaminophen is the most common cause in the U.S. today and accounts for approximately half of all cases of acute liver injury in our country each year. However, diagnostic tests to evaluate acetaminophen liver injury are inadequate and only work in the first 24 hours after an acetaminophen overdose. Our laboratory has established a rapid and new assay to detect acetaminophen liver injury. Years of research in animal models have established that acetaminophen is metabolized in the liver through an oxidation process that generates a highly reactive metabolite. This metabolite binds to cysteine groups on protein, forming acetaminophen protein adducts, or APAP-Cis. As these products are formed in liver cells, the liver cells rupture and burst and release their contents, including acetaminophen protein adducts, into patient blood. For the last decade, our laboratory at Arkansas Children's Research Institute and the University of Arkansas for Medical Sciences has quantified the levels of acetaminophen protein adducts in patient blood samples over a range of acetaminophen exposures. We have also determined the level of adducts that correlate with high levels of serum ALT, a common clinical indicator of liver injury. The HPLC-EC assay has allowed us to quantify levels of acetaminophen protein adducts. The study published in the April issue compares a rapid assay for qualitative measurement of acetaminophen protein adducts to our quantitative HPLC-EC assay. Acetastat, the rapid assay, performed very well and generated the same results as the HPLC-EC assay for acetaminophen protein adducts. 81 patients participated in the study, including 33 with acetaminophen liver injury and 29 with non-acetaminophen liver injury. Acetastat, the rapid assay, gave the same results as the HPLC-EC assay in 95% of the patients. These data are highly encouraging and suggest that acetastat may be a very helpful diagnostic assay to be used in the future to evaluate patients with liver injury. Because acetastat is a competitive immunoassay, the disappearance of a line at the test band indicates that the patient's sample contains acetaminophen protein adducts and is thus a positive test. A positive line at the control band indicates that the assay is working.